Hi everyone, how are you all? So myself Dr. Amit Jain, I'm your faculty of biochemistry at Allen Next. And guys, as we all know, the INI-CET is just a few days away, right? Or you can say 2 to 3 weeks, 3 weeks away, right? And we all know that first year and second year subjects are very important for especially the INI-CET. And in this rapid revision session, what I will be doing is, I'll be taking the genetics, molecular genetics part. From the biochemistry session, I'll be taking the molecular genetics part. So that is very, very important topic. See guys, in INI-CET or in the NEET PG, most of the time what we are getting is the repeat PYTs, okay, previous year topics, and maybe sometimes directly the previous year question, right? So question may not be helpful always. See, I know there is a question always on some topics like, let's say, mRNA editing, but that question may be changed, okay? It has been asked in aims like previously, like the name of the enzyme which causes mRNA editing. There are questions on microRNA, so many times they have asked similar questions also and different questions also. So what we should know is, we should know the previous year topics very well and not only the previous year question, okay? So in this topic, in this session, I'll be taking the genetics section, which is holding a very much importance in the INI-CET exam especially, right? So let's begin without wasting time, and if you have any question, keep asking me, right? I'll be here to answer all of them, right? So let's start. This will be your session like which will be game changing. This will be something which I'll be giving everything in a concise form from the genetics section. You know, normally when you see the video of genetics section, it will be almost of 10 hours, right? So I'll be trying to cover this in the very less time, whatever it is possible. So let's start with the slide number 1. Yeah, hi, good evening everyone. So let's start, okay. So guys, let's begin with our first topic, and that is Human Genome Project. So recently there was a question on Human Genome Project. Okay, so what we should know, the points they asked is like which of the following is correct and which of the following is incorrect regarding the Human Genome Project. Such was the question in one of the recent exam. So it was launched in 1990 and it was completed in 2003. Okay, what was the goal? The goal was to analyze, right, the human genome, right, to find the location of genes and to create a map. Okay, what are the steps? DNA of the cell is isolated and randomly broken into the pieces using the DNA knife. So what is the name of the DNA knife we have? That is restriction endonuclease. So that was used. Okay, then inserted into the vectors. So what were the vectors used? In this case, the vectors used was bacterial artificial chromosomes and the yeast artificial chromosome. So these were the vectors which were used because they can carry the larger amount of the DNA. That's why. And then they are cloned into the host. Okay, then they are cloned into the host like the bacteria. So guys, PCR was used to make the copies of the the DNA that was cloned. Fine. Okay. Then we have Sanger sequencing. So what was the technique used? Very important to remember, that was Sanger's sequencing. Okay, that was a topic that was used here. Fine guys. So you should know that it was not the next generation sequencing. It was the Sanger's sequencing which was used in this process. You got my point? Not the next generation sequencing. And then the sequences are arranged, right, and assigned to a chromosome, and the maps are constructed, right? What are the findings? Let's see what are the findings of the Human Genome Project. So human genome has 3 billion base pairs, we all know, right? And there are around 30,000 genes. Each gene has around 3,000 base pair of size. Now this was again a recent point, right, that dystrophin gene is the largest, that is already being asked, and this was a recent one, that is SRY, or the testis determining factor on the gene on the Y chromosome, that is the smallest gene. That was a recent MCQ asked, right? Okay, then moving to the next point, proteins are coded. So this question has been repeatedly asked previously in AIMS and NEET PG also, that how much percentage of the human genome is coding DNA. So how much is the coding DNA? Less than 2%, or precisely if I say it is less than 1.5%. Okay, around 1.3 to 1.5% is coding DNA. So guys, that means what? The most of our DNA, human DNA, is non-coding, right? Or it is having some repetitive sequences. Okay, and that is what you call, as a satellite DNA. Fine. So chromosome 1 has the most genes, and the chromosome number Y has the smallest number of the genes. Okay, repeated sequences are present where? I told you, like, repeated sequences, they are called as satellite DNA, and they are mostly present in the centromere and the telomere. Fine. Is it correct? Okay guys, let's start with now DNA. So we will be taking the sections like DNA, RNAs, and regulation of gene expression, some points on translation and recombinant DNA. So I'll be covering these 5 topics, right? And they are very, very important topics. Okay, so high yield points or the previous year topics and the questions that I'll be covering here, right? So let's begin again. So guys, when we talk about the DNA, what are the basic things that we should remember? That we all know it's a double-stranded anti-parallel right-handed helix. Okay, and the base stacking and the hydrogen bondings are the main forces, right? And Chargaff rule says that A plus G, that is the purines, are equals to pyrimidines, that we all know. So Chargaff rule is very important, guys. It is again one of the PYT, on which, like, you're being asked questions on calculate if A is given 10%, how much is the C, like that. Okay, so if A is 10, then T must be 10, so like that. Then guys, hydrogen bonds between the G and C, there are triple hydrogen bonds, and that is why the DNA which is rich in G and C has a higher melting temperature, right? This point was again recently being there in the MCQ, and that was a correct statement regarding the DNA. That is, G and C rich DNA has the higher melting temperature. Fine. Guys, single-stranded DNA, like during PCR, what happens? The double-stranded DNA converts into 2 single strands. So how do we know that the DNA has separated? So guys, single-stranded DNA absorbs light at 260 nanometers. So this is called as hyperchromicity of DNA denaturation. क्या बोलते हैं? Hyperchromicity of DNA denaturation. So that is at 260 nanometers, right? Okay. Now let's talk about the DNA packaging. So DNA is negatively charged and binds with the lysine and arginine rich histones. So guys, we all know the DNA is a thread and it is wrapped over the histone proteins. Okay, and this is called as nucleosome. Now there is something called as histone octamer. Histone octamer has all the histones, that is A, B, right? H2A, H2B. Fine, but there is H3 and H4, but there is no H1. So that has been again one of the MCQ, right? That has been asked as one of the MCQ also. Fine. And guys, you should know that around 146 base pair of the DNA is wrapped over the histone octamer in just 1.75 turns. How much turns? 1.75 turns. So nowadays, though they are not asking any data-based question directly, है ना, वो directly नहीं पूछते कि भाई ये कितना होता है या most common क्या होता है. ये questions अब थोड़े कम हो गए हैं. Like these are the less frequently asked questions nowadays. But yes, has been asked, so that's why I have covered it. You should know this, right? Okay. So guys, this is when the DNA is being packed, you know, under the electron microscope, it gives the beads on the string appearance, right, under the electron microscope. So this can be that you know, image that you should know here. Guys, DNA replication, again the basic things, that is about the prokaryotes. What we should know? We should know, like, because in the INI-CET exams we will get questions like, you know, multiple correct maybe, or maybe the questions which there is a sequence, ascending order, descending order, etc., right? So what is the sequence of enzymes in the prokaryotic replication? Okay. So guys, the sequence of enzymes in the prokaryotic replication is, see, first of all, we have to separate helicase. Second is topoisomerase, which will remove the supercoiling, right? Then third, the DNA polymerase. First of all, then RNA primase is there. So primase is making the small RNA primer, because the DNA polymerase cannot work without a pre-existing 3' OH group. So we have to add it first using the RNA polymerase, that is RNA primase. Then we will use the DNA polymerase 3 and the 1, and finally the DNA ligase. So 3, 1 and ligase. So guys, very basic question. See, the good thing about the biochemistry subject is that questions are mostly from the PYTs, right? Rarely there are new questions. Okay, so like DNA replication is one of the important PYT, and it is very basic that we should always revise it. See, our goal is to revise the subject in the last 7 days, especially the first prof and second prof subjects are very commonly asked in exams in the INI-CET. So what should be our goal? That in the last week of the exam, if we are able to revise it, then we are prepared, right? Then we will be able to get or achieve the rank that you are desiring for, right? Okay, ठीक है, चलो, let's move ahead. So activity found in DNA polymerase 1 but not in DNA polymerase 3 and clino fragment. So can you answer this question? What is the activity found in DNA polymerase 1 but not in the 3? So guys, that is an activity to remove the primer. And what is that activity? That is the 5' to 3' exonuclease. Okay. So what is clino fragment? Clino fragment is again the DNA polymerase 1 which has, which is not having this particular activity. Again, a very important PYQ has been asked several times in the previous exams. Remember, the proofreading is carried by the DNA polymerase 1 and 3 in the 3' to 5' exonuclease activity. Guys, remember that proofreading is one of the only activity in the genetics which is in 3 to 5 direction. This was again the question was asked in AIMS previously, that there is which of the following process occurs in 3 to 5 direction, and the proofreading is the answer to that. Right? Most of the things we will be talking about 5 to 3, like 5 to 3 polymerase, 5 to 3 exonuclease, okay, for the primer removal, and then 3 to 5 exonuclease for proofreading. What is proofreading? That is removal of the mistakes. Okay, let's move to the next point. So guys, eukaryotic DNA polymerases, we should know them. These are alpha, beta, gamma, delta and epsilon, and gamma is especially important for the mitochondrial DNA synthesis, right? And beta is required for the base excision repair, has been asked in the PYQ, right? Rest are delta and epsilon. Fine guys, so these are the important DNA polymerases. Not frequent one, but yes, you should know. So guys, this is again the same figure that we have already discussed. Okay, that is helicase is moving the first, and then there are other enzymes which are acting, like topoisomerase is acting the second. Okay, or you can say actually simultaneously, right? But first mark with helicase. Guys, telomerase is again a super important, very important topic that is frequently asked in AIMS and INI-CET exams. Right, so all the central institutes have asked about telomerase enzymes. So very important topic, and you must know everything about it. Okay, so I'll be telling you some most important points. So this is called as RNA dependent DNA polymerase, or also called as a reverse transcriptase. Clear? Then guys, telomere, the end of the chromosome, has the repeats which is AGGT, or you can say AGG repeats. Fine. Where is the telomerase enzyme which maintains the length of the ends of chromosome, right? So where it is present? So it is present in stem cells and guys, male germ cells, right, or the male sex cells, right, which are creating the sperms. But female cells have very low activity. Then cancer cells get them activated by the various processes, right? So remember these three important points, that where is the telomerase enzyme is present in. Fine. Now guys, there was a question asked that what is the Hayflick limit? What do you mean by the Hayflick limit? So guys, Hayflick limit is that every cell in our body is able to divide only for the 50 to 70 times, right? And guys, this will be then going to apoptosis. Why? So if the next question is that why there is a Hayflick limit and why the cell goes to apoptosis after a certain number of divisions, so the answer is because it is dependent on the length of telomeres. Okay, so if the length of the DNA shortens, then obviously there will be what? There will be the senescence or the death of the cell, and that is called as Hayflick limit. Very important, this question has been asked. Okay, then moving to our next point. Guys, this question, though it has not been asked, but I'm just telling you the dysfunctional telomerase gene causes a disease known as the dyskeratosis congenita. Okay, and this disease presents with a triad of, as you can see, the nail dysplasia, skin pigmentation or the reticulated skin, okay, mash-like skin, and the oral leukoplakia. Fine, so these are the three points that we should remember about the disease known as dyskeratosis congenita. You got my point? Let's move to the next topic, next point. Guys, again a very, very important topic, right? One of the favorite of examiner, okay? Though they have asked most of the time very easy and superficial questions from this topic, but still we are creating mistakes in the exams, okay? So that should not be there. Till now they have asked mostly about the questions about the name of the disease and the defective DNA repair. But what we should know is DNA damage, right, and how it is being repaired. So guys, first of all, if we see the reactive oxygen species and the ionizing radiation like X-rays, okay. So guys, what they do is they all causes one thing, and that is called as a basic site, means remove the base or the single strand break. And guys, how to repair it? So we can repair it by the base excision repair. Remember this. This can be repaired by base excision repair. So A basic sites are repaired by base excision repair, and they are created. What is the source of the DNA damage? As you can see again, reactive oxygen species, okay, ROS. So remember ROS, like superoxide for example, okay, or anything else, that is causing the formation of A basic site. So this can be the MCQ in your exams, future exams, though it has not directly been asked till now. Then guys, we all know UV rays. What UV rays do? UV rays causes the formation of thymine dimers, okay. They are causing the formation of thymine dimers, T dimers, okay, or you can also call them pyrimidine dimers, and this is being repaired by the nucleotide excision repair. Very common question has been asked previously. UV rays damage, thymine dimers repaired by nucleotide excision repair, okay. Moving to the next point, guys, again the ionizing radiation, they can cause the DNA double strand breaks more commonly than the single strand breaks. Though I have written here the single strand breaks also, but that is mainly by the point of reactive oxygen species, okay. So double strand breaks are repaired by the process that is called as double strand break repair. And guys, double strand break repair is more commonly done by a process that is called as non-homologous and joining, right? Non-homologous and joining compared to homologous repair, right? I'll tell you why, reason being that, but just remember it is more commonly by the NHEJ, right? Non-homologous and joining. Fine. So, and the last is replication errors. Replication errors, guys, like insertion, deletion or the base mismatches, they are being repaired by the mismatch repair. So guys, though we all know already, we have we have revised the names of disorders, but these can be the questions, right? Like already they have asked thymine dimers multiple times, but they can ask you other questions also, like how will you repair the damage caused by reactive oxygen species, for example? How will you repair the X-ray cause, suppose the double strand breaks in the DNA, how will you repair that? What will be the mechanism? Okay, so that will be like, let's say, the double strand break repair. So don't forget this table. This can be very important table for the future exams, right, in this exam. Okay, so let's move to our next point. So guys, I'm just talking about the names of disorders that we already know. So first is HNPCC. It is caused by the defective mismatch repair. Fine. Defective mismatch repair. Guys, remember the mismatch repair machinery may promote the trinucleotide repeat expansion. Okay, so mismatches can be dangerous because they can promote the expansion of trinucleotide repeats. Like for example, we all know in some disorders there are trinucleotide repeat expansion, for example in Fragile X syndrome, for example. Clear? Then moving to the next point, okay, we have xeroderma pigmentosum, cocaine syndrome. Okay, xeroderma pigmentosum, cocaine syndrome and trichothiodystrophy. So guys, this is being repaired by, or it is defective nucleotide excision repair, right? Okay, next is the so defective nucleotide excision repair. Guys, xeroderma pigmentosum has increased risk of skin cancer, right? Which skin cancer? Most commonly the basal cell carcinoma is most common. And in the Cockayne syndrome, there is no risk of skin cancer, but the patient has a face which looks like, the bird-like face. I'll show you the image. Then pibidis, that is trichothiodystrophy, is also called as pibidis syndrome, which has this full form, right? You can, if you want to remember. But this was a question which was asked in exam, that is tiger tail banding, right, is noticeable under the polarized light microscopy. So tiger tail banding of the hair, see the name, trichothiodystrophy. Tricho means hair, thio means sulfur deficient, thio means sulfur, okay, and dystrophy. So sulfur deficient hairs. I'll show you the image again. Then guys, though as such not a direct disease name here, but this was a question asked that which of the following enzyme is utilized in the case of base excision repair. Okay, so which of the main enzyme of base excision repair? So what is a base excision? Like if there is a wrong base like uracil in the DNA, how will you repair it? To repair the uracil, to remove the uracil, we need enzyme that is called as glycosylase. Okay, and if this glycosylase or MUTYH is defective, then this can increase the risk of colon cancer, right? Okay, guys, remember one more thing that I told you previously, that reactive oxygen species, what they do is they causes a DNA damage. Fine, they cause a DNA damage. What is the product formed by the ROS damage? Guys, the name of that product is called as 8-oxo-G. That is, remember just the deoxyguanine. Okay, remember just name one name, that what is the marker of DNA damage by the reactive oxygen species? The answer will be deoxyguanine. Correct? Fine. So let's move to the next point. So Werner syndrome and Bloom syndrome, very commonly asked till now. What is the defect? The answer is helicase enzyme. Werner syndrome and Bloom syndrome, they present with multiple cancers, okay, and defective helicase, right? So multiple cancers and increased or the faster aging, right guys? Then moving to the next point, hereditary breast and ovarian cancer, the risk of hereditary breast, ovarian and the prostate cancer, that is because of the defective homologous repair, right guys? So homologous repair, that is done by the BRCA1 and BRCA2 proteins are required there. SCID is caused by the defective non-homologous AND joining. This happens due to the defective immunoglobulin gene process during which the immunoglobulin gene has been targeted, right? Which process? That is a antibody diversity generation, right? So during that's a physiological process which needs the immunoglobulin gene to be broken, that is double strand breaks, and that is repaired normally by NHEJ, non-homologous AND joining. So this has been asked again 2 times in the exams. Which question? That is SCID, severe combined immunodeficiency, right? Now guys, there can be a new question. Let's take that point. Why the non-homologous AND joining is preferred over the homologous repair, okay, during the DNA breaks or double strand DNA break repair? Why we choose non-homologous, which is prone to more mutation compared to the homologous repair? Guys, the answer is simple, because the heterochromatin, that is a DNA which is tightly bound with the histones, that is DNA which is non-accessible, right? So if the DNA is non-accessible, then we cannot take the the sequence for the homologous repair, because we need the sequence for homologous repair. So that is why we prefer NHEJ. So that's why you have to mark NHEJ as the best answer. That has been asked twice in exams, okay? And one more question, which phase of cell cycle is most radio resistant? This was again the recent last 1 or 1.5 years back question. So guys, S phase of the cell cycle is the most radio resistant. Which phase? S phase, okay, synthetic phase. Why? So why it is radio resistant? Again the same reason guys, because that is the time the homologous repair, which is error free, will be active, because the DNA replication is going on, the DNA is loose, right? That is, there is euchromatin, and the DNA and the proteins which are required for the DNA repair, they can access the sequences required to repair or replace the lost part of the gene. That is why. So these two questions can be again they can be repeated. This was recently repeated. Which phase of cell cycle is most radio resistant? Okay, fine. So let's move to our next point. Okay, that is just images has been shown. So guys, this is a case of you can see this is a case that is of trichothiodystrophy. Okay, and especially this point, that is tiger tail appearance or the tiger tail banding of the hair, right? And this is in the right hand side, we have the xeroderma pigmentosum with the pigmentation, the dryness, and the these are the areas where the cancer has been shown, right? Like the basal cell carcinoma, what is the common site? The common site is the nose. Okay, and this is a case, as you can see here, this is a case of trichothiodystrophy, right, where the the face has a typical appearance like a diseased bird. Fine. Okay, now guys, can you tell me the names of disorders which are caused by the defective proteins, right, DNA repair proteins like helicase, nucleotide excision repair, and the laminin genes. So these are the names. Okay, the helicase, the defect in the helicase causes Werner, Bloom, and Rothman-Thomson syndrome, and the defect in the nucleotide excision repair causes cocaine, xeroderma, and trichothiodystrophy, as I told you already. And defect in the laminin proteins causes the disease which is called as Hutchinson-Gilford progeria syndrome. So that is, these are all the disorders which are sun sensitive and with increased aging process, faster aging process, right? Okay, though they don't survive beyond certain age, and they have a faster aging process. So these are the disorders. As per the color, you can answer it. Helicase, red color, and that is rest of the colors. Fine. So let's move to our next point. Guys, recently there was a question on the structure of genes, that is enhancers and silencers. They asked some points like which of the following is correct regarding the eukaryotic genes, or some points on that. And as per the recall base, if we say that. So this is the structure of the gene, right? We all know that in the gene there are some promoters, right? Then there are some probably, if you don't know, that is called as a locus control region, insulator, silencer, and enhancer, right? So these are the components of the gene: promoters, locus control region, silencer, and enhancer. So that is a eukaryotic gene I am talking right now. Okay. Now guys, what is locus control region? Locus control regions are the group of transcription regulatory elements, right? And these are present, okay, these are present especially in the genes, right, which are important for cellular differentiation. Like for example, we have the genes of the beta globin, so beta globin genes, right, and other genes are like MHC genes, right? So these are the genes. Then there was a question on enhancers, that where they are present. Let's talk about enhancers and silencers. So guys, enhancers, they don't have any fixed location. That you should know, that they are independent of any direction, right? So enhancers may be located 5' or 3', that is upstream or downstream of the gene they are affecting. Enhancer means they increase the rate of transcription, make more mRNA, and that from we will be getting more of the protein, right? So that is enhancing the transcription rate. Second point is that they are not located near the transcription initiation site. Okay, so they are located, may be located several thousand of bases away from the transcription initiation site or the promoter. They are not directly affecting the promoters also. So they do not interact or do not act on the promoter region itself, right? But we need some mediator proteins. So 3 points again. They are located maybe upstream or downstream of the gene they are affecting. Second thing, they may be present anywhere, maybe near or thousand of the bases away from the gene they are affecting, and they don't directly bind with promoters, but they need a mediator protein. So these 3 points you must know about the answers. This question has been asked till now only once, right? So that can be repeated again. तो यार ये repeat question गलत नहीं होना चाहिए because that will be counted as a PYT. तो हमारा point ये है कि हमें PYQ करना है ना, और PYQ डायरेक्ट आता नहीं है मोस्ट ऑफ द टाइम, उससे रिलेटेड क्वेश्चन आते हैं, राइट? तो हमारा PYT बन जाता है वो, तो ये PYT है एक तरह से आप, क्योंकि एक बार आ चुका है ये, राइट? एंड दैट बिकम्स इंपॉर्टेंट. एंड लोकस कंट्रोल रीजन मैंने आपको बता दिया क्या होता है. सो ये स्पेशली जीन्स में होता है जो सेलुलर डिफरेंशिएशन में या विच आर रिक्वायर्ड ड्यूरिंग द developmental stages like MHC and the beta globin genes have LCR. Just for your understanding, guys, as you can see, this is the enhancer, right, and of the gene, and this is called as a promoter. Pink color is promoter. This is a promoter. Black color is enhancer. And these all the proteins, they help in their binding to each other, right? So how does the enhancer activates the transcription of a gene? By some mediator proteins that we are not concerned with right now. Okay, guys, so that is how the enhancer may be present anywhere, as I told you, right? See, the gene 1 is actually being affected by this enhancer, fine, but it is present somewhere between gene 2 and gene 3. So as you can see here, right, the gene 1, the real target gene is gene 1. So guys, don't forget this point. This will be a very important question in the exams, right? Can be repeated again because it was a recent MCQ. Okay. See, genetics, like in the INI-CET probably you will be getting questions which are almost from, in the biochemistry you will be getting around 10 questions or 10 to 12 questions, right? And from them, there are sometimes a lot of questions are from the genetics section also. Okay, though some topics like we all know vitamins and amino acid ureas, and the disorders of glycogen or the lipid metabolism, they are also being asked frequently, but genetics holds always an important place, and that's why I made this video to make it most high yield, right? So let's start with the next point. Guys, RNA polymerases of the eukaryotes. So there are 3 RNA polymerases of the eukaryotes. RNA polymerase 1. Guys, RNA polymerase 1 makes the ribosomal RNA. But there are 4 types of ribosomal RNAs, 28, 18 and 5.8. But 5 also, which is not made by type 1 but by the type 3. That was MCQ in one of the exams. RNA polymerase 2 makes microRNA, very important, and definitely we know messenger RNA, right? Messenger RNAs and microRNAs. And very important, guys, this can be the question because it has not been asked much in exams, especially the INI-CET. The alpha amanitin is a mushroom poison which inhibits RNA polymerase 2, and it has no antidote, right? It is derived from some mushroom, that is alpha amanitin. It's a mushroom poison. Guys, poisons and inhibitors are always important for exams like INI-CET or NEET PG. And we have in our subject biochemistry very few numbers of names of poisons and inhibitors, and they are very frequent. Like for example, if I say electron transport chain and other topics, they are very frequently asking the inhibitors of complexes. That should be on your tips, fine. But today I'll be telling you hardly 2 or 3 names of inhibitors that has not been asked more commonly, so they can be asked again, right? So that is your alpha amanitin and mushroom poison, right, guys? Guys, remember one thing, that the RNA polymerase of the eukaryotes doesn't bind directly to the promoters, right? This is very important line, and it can be the question in the next exams, right? The basic knowledge question, that RNA polymerase of the eukaryotes doesn't bind directly to the promoters and needs some transcription factors or mediators like TF2D and TF2F, okay? So that you should remember. RNA polymerase 3 is making tRNA, 3 for tRNA and 5S RNA. This was a question in the previous exam question, PYQ. Not necessarily INI-CET, I mean to say like AIIMS or the Central Institute exams also. INI-CET started only a few years back. So definitely this question can be asked again. 3 is making also the ribosomal RNA, 5S ribosomal RNA. Don't miss that. These points if we miss, we will not be able to get the PYQs correct, okay? PYT is correct. So my point is, guys, कि PYT गलत नहीं होना चाहिए, है ना? टॉपिक गलत, टॉपिक वो अच्छे से आना चाहिए जो पहले आ चुका है, फाइन. Chalo, let's move to the next. Prokaryotes have a single RNA polymerase and it has multiple subunits, out of which sigma is the one which identifies the gene or the promoter and helps in initiation of transcription. And guys, it has also some beta subunit which is inhibited by the antibacterial rifampicin. You can remember that RNA polymerase of bacteria is inhibited by which antibiotic? Rifampicin. Rho factor, which is not a part of RNA polymerase, it is different. See, this is not the part of RNA polymerase, not the part of RNA polymerase. Okay, this is the enzyme which releases the mRNA when it is being synthesized. Let me show you just a picture of that. So see guys, sigma subunit is binding the DNA, not the other subunit. So sigma is the most important for transcription initiation, right? And as you can see again here, I told you that the Tata box, or I mean the promoters of eukaryotes, is not directly bound by RNA polymerase. First we need some, transcription factors like TFD, 2D, 2 for RNA polymerase 2. So RNA polymerase comes later. First there is a binding of some transcription factors like TF2D, and F is important because it takes the RNA polymerase. Okay, D and F. F is not shown here directly, but D is being shown here. F is the flight of RNA polymerase to the gene, right? Okay. So let's move to our next point. So this is just an image of the, guys, the mushroom that is called as a death cap. Fine, it is a cap and it is called as a death cap, right? It has alpha amanitin. It is also called as a destroying angel. And now wo kya hai? I don't know that. Fine, let's move. Angels are not supposed to be destroying. So guys, there are 3 examples of once the RNA is made in the human beings or eukaryotes, there are 3 post-transcriptional modifications. Number 1, we add a cap that is called as 7-methylguanosine. It is added to the first nucleotide of the RNA by a bond which is not the phosphodiester bond, 5' to 3' phosphodiester bond or anything, but it is a triphosphate bond, right? So guys, remember, mitochondrial mRNA has no cap, MCQ. Mitochondrial mRNA has no 7-methylguanosine cap. Then the next point is, we have the next point, that is functions of the cap. Okay, what are the functions of the cap? So number 1, protect from the cleavage, right? And then unique identifier of the recruiting protein factors for the pre-mRNA splicing and poly-A tailing and the export from the nucleus. Guys, the most important point is that I have highlighted that it is important for the initiation of translation. I'll show you that cap identifies the 4E, initiation factor for protein synthesis. So if the mRNA has no cap, no tail, and if it is not spliced, it will not be utilized for making the proteins. How to identify mRNA which is being ready for the protein synthesis. तो उसके पास कैप होना चाहिए, टेल होना चाहिए और वो स्प्लाइसिंग होना चाहिए. मतलब कि हमें उसका एक्सॉन्स को ज्वाइन करना पड़ेगा, इंट्रॉन्स को रिमूव करना पड़ेगा. तो ये आपने पढ़ा ही है सबने पहले. You all know these points, right? I'm just highlighting that what are the important points that you should remember. If these points are not in your revision, then you are missing a lot, right? Okay, so this is the cap. Then guys, 3' is we are adding the tail, right? And the tail is the site of binding of some proteins known as poly-A binding proteins. So guys, that is important for identifying again the mRNA which is processed, which is ready for translation. Okay, then what is pseudo-circular structure that I'll show you? Removal of the non-coding sequence or introns, non-coding sequences. If you remove, that is called as the splicing, and so we will join the coding sequences known as exons. Clear? So guys, this is catalyzed by the SNRP, SNRP proteins, which contains a ribozyme known as snRNA or the small nuclear RNA. So examples of ribozyme that we have to remember, and this is one of the example of ribozyme. Clear guys? So what are the names of SNRP proteins? These are uracil rich RNAs. So they are having U1, U2, U4, 5 and 6, but not the U3. Once in the aims they asked that U3 is required for SNRP is required for which of the following purpose, and the answer was it is required for ribosomal RNA synthesis, but not for the mRNA splicing. Okay, it is not required for mRNA splicing. Remember this PYQ it is, right? If it is not in your notes, please add it. Okay. So guys, these are the three important processes which are called as post-transcriptional processes, processing of mRNA, right? Modifications of mRNA. And guys, remember there was a question asked that which of the following disease is example of splice site mutation? Splicing, we have to cut the introns out. So, these sites have specific sequences, and if these sequences are mutated, then the introns will stay in the mRNA, and that is the example of, like for example, beta thalassemia is one of the example of the disease splice site mutation, PYQ again, right? Name the disease of splice site mutation, that is beta thalassemia. Let's move to the next point. So guys, again I am just showing you here that this is the cap, this is the tail, poly A tail, and when the protein is being synthesized, this cap is bound with certain factors like 4E and etc. And this tail is bound with poly A binding protein and 4G, okay, and the 4G. So that will be covered again, don't worry. So this is your mRNA and it becomes pseudo-circular. So during the protein synthesis, the mRNA is being pseudo-circularized. That is just a term that you should know. Okay, so guys, again, capping, tailing, splicing I have covered, and all the points I have covered there. And this is again the same picture, right? Cap is bound by some proteins, initiation factors, and again the same poly A tail is bound by the poly A binding proteins. So let's move to the next point. That is again just a figure that is showing how the introns are being removed, so we all know that. Now guys, let's start with the most important topic in INI-CET, what I believe is that is the regulation of gene expression. Okay. So this is one of the most important topic, regulation of gene expression. First in the prokaryotes. So guys, in the prokaryotes we have a topic that is called as a lac operon. So in the lac operon, what we should know is that in the lac operon what we are talking is that how does the bacteria controls the genes which are making the enzymes to digest lactose. Okay, so to digest the lactose they have three genes, and the three genes are called as ZYA. ZYA, three genes are there, and these three genes are controlled by a single promoter. Okay, in human beings or eukaryotes we have one gene and one promoter. Here there are three genes which are making the enzymes to digest lactose and one promoter. So guys, this concept is called as operon concept, when the multiple genes are controlled by a single promoter. Okay, and they have a site that is operator, which is not found in our genes. Okay, but only in the prokaryotes. So not in the eukaryotes, operator region is not found. Fine. So guys, what we have to remember in this case is that there are a lot of things written here, but what we have to remember is is one thing, that is okay, I'm just looking at the chat here, and just a second. अरे राहुल, ये लाइव नहीं जा रहा क्या यार? वो तो वापस से शुरू हो गया मेरा. वापस से शुरू हो गया ये तो. नहीं सर, चलो. तो उधर क्यों नहीं जा रहा? मैं कंफ्यूज्ड हो गया यार. Okay, so in this topic, what I just want to tell you is that in the lac operon, what we need to remember, that lactose is not the preferred choice of the sugar which is utilized by the bacteria, right? Lactose is not the preferred sugar, it is the glucose. So guys, when the bacteria will make the lactose digesting enzymes, Answer is when the glucose is absent and the lactose is present. You got my point? When the glucose is absent and the lactose is present. So that is the only condition where the bacteria makes the lactose digesting enzymes, or I can say the lac operon is expressed when the glucose is absent and lactose is present. So guys, when the glucose is absent, the bacteria is under the stress and there is increase in the levels of cyclic AMP and the cap proteins. So this cyclic AMP and cap, they bind, as you can see here, cyclic AMP and cap, they promotes the RNA polymerase binding to the lac operon, and that's how the lac operon is expressed. Again, I'm telling you, when the lac operon is expressed, glucose absent, lactose present. When the glucose is absent, there is rise in cyclic AMP and the cap, and they promotes the RNA polymerase binding or action of RNA polymerase, that is the transcription of the genes, okay, to make mRNA and then to make the proteins. So that is very easy, guys, nothing difficult here. Just remember this line and you're done with your MCQ. So I can tell you just one thing, that if I talk about the lac operon, then what you should know is that glucose present and lactose is absent. Then guys, there is a protein known as repressor, right? And repressor doesn't allow the RNA polymerase to bind. So as you can see in this picture, this is repressor. And guys, repressor blocks the RNA polymerase activity. So repressor blocks and cyclic AMP allows the activation of lac operon. You got my point? Just these 2 points. Repressor block, and when is a repressor present? Active when there is glucose is present. If glucose is present, bacteria will use the glucose, that's it. So let's move further. So glucose present, lactose absent, R will be considered as a negative regulator of lac operon. Okay, glucose is absent, this is the condition that is required, lactose present. So then lactose binds the R and inactivates the operator, right? So operator is free and cyclic AMP levels rise and the cap binds, the RNA polymerase is activated, and that's how the lac operon is expressed. So guys, cyclic AMP cap is the positive regulator. This was MCQ, PYQ. So these are the 2 PYQs from this topic till now. Glucose and lactose both present. So it's simple, bacteria will use the glucose and not the lactose, right? But there will be one thing, that the lactose binds the repressor and the operator is free. So lactose inactivates the repressor. Fine. If you want to see this again, let me show you. Fine. As you can see here, that repressor binds operator and it blocks the RNA polymerase activity, right? It binds here, okay, and it will block the RNA polymerase activity. Fine. Just a second, guys. Okay, so this is what we have all discussed, and we were at lac operon. So that's it, guys, about the lac operon. Now let's talk about the eukaryotic gene expression. Next topic, eukaryotic gene expression. So guys, eukaryotic gene expression is again one of the very important topics, which is highly repeat MCQs. So I will be covering them in these 5 subtopics, though not all the 5 subtopics are very important, but only points which are high yield or which I believe can be MCQs in next exam, I have just taken them. So let's start with the epigenetics. So guys, epigenetics control. Epigenetic means, above the sequence of DNA. We are not changing the DNA sequence. So number 1 is, there are 3 mechanisms of epigenetics. First, you should know the name of 3 mechanisms: DNA methylation, histone modification. Number 2 is histone modification. Number 3 is microRNA or the gene knockdown. Okay. First is DNA methylation. So guys, where we do the methylation? At the CpG islands, right? And this is causing the silencing of the gene. Methylation means silencing of the gene. Where we do it? At the cytosine of the CG-rich regions, which may be near to the promoters. Okay. And guys, this can be cancerogenesis because it may inactivate the tumors suppressor genes, right? Next is histone modification. Like guys, there are a lot of histone modifications are there, but acetylation of the lysine is the one which activates the gene. Acetylation activates, and deacetylation inactivates the gene. Clear? And there are some drugs, right, like vorinostat, which inhibits the histone acetyl transferase, right? Okay, so this is like histone acetyl transferase, fine, and these drugs are used in the treatment of some tumors, right? Next is RNAi or the gene knockdown. So guys, this is one of the very important topic again. RNAi for RNA interference, okay, and the gene knockdown. So microRNA, which is 21 nucleotide long, right, binds to the 3' UTR of the target mRNA. So there is a small RNA that is made by our own cells, right, which is just 21 nucleotide long. Where does it bind? This was MCQ, 3' UTR, untranslated region of mRNA. So guys, this is very important MCQ, that what is the binding site of microRNAs. So 3' UTR of target mRNA, and this inhibits the translation or maybe degrade the target mRNA, you know, right? So jo bhi mRNA which is causing, let's say that there is one mRNA which is causing excess production of the growth factors, so that may cause cancer, right? So our body has a mechanism to prevent the making of these growth factors, suppose via inhibiting that mRNA or degrading that mRNA using this small microRNAs. This is called as interfering the function of RNA. That's why RNAi, interfering with the function of RNA, right? And RNAi involves, guys, RISC, that is RNA induced silencing complex. So once they ask the question that what are the components of RNA induced silencing complex. So these are the proteins, that is AGO2 or the slicer endonuclease. What is that called as? Slicer endonuclease and the dicer. So these are the two names which are part of the RISC, RNA induced silencing complex. Obviously the one of the component is microRNA. Clear, guys? So that is your epigenetic control. Fine. Now let's talk about the transcriptional control. So guys, in the transcriptional control we have promoters like Tata box and TF2D I told you in the beginning. So that is the control of transcription, that how the transcription factors are helping. I have already talked about enhancers and silencers, right? So there is nothing new here. We have already talked about them. Then post-transcriptional control, that after the transcription is over. So guys, this is called as alternative splicing. What is alternative splicing? That we have more than 1 lakh proteins, but from just, let's say, 30,000 genes. So how come we have more than 1 lakh proteins from 30,000? It should be 30,000 protein only ना, सिर्फ 30,000 होने चाहिए थे, but वो बहुत ज़्यादा है. वो क्यों है? Because we are able to create multiple copies of the mRNA, right? We are able to create the multiple copies of the mRNA, right, from the one mRNA. That is via the splicing, that is alternative splicing. Okay, fine. So guys, that is the meaning of alternative splicing. Like we have the number of neuraxin proteins from the only 3 neuraxin genes. Okay, and the B lymphocytes produce both cell-bound and the secreted antibodies. That was again the question which was asked in the previous exams, right? That how does the B lymphocytes are able to produce the antibodies which are cell-bound and secreted also. So that is through the process called as alternative splicing. Because during the alternative splicing, we will remove one of the exons that is coding for, for the, what I can say, the amino acid sequence which is binding the protein to the cell membrane. तो हम वो एक्सॉन ही हटा देंगे जिससे वो सेल सरफेस पे बाइंड करता है, सीक्रेट हो जाएगा वो. क्लियर? नेक्स्ट इज, his next point is that mRNA stability. See guys, let's take the example of iron deficiency anemia, and in the iron deficiency anemia I want the ferritin mRNA to be degraded, but the transferrin receptor mRNA to be stabilized. So I don't want ferritin, storage of iron protein, but I want transferrin receptor. So I have to stabilize the transferrin receptor. Okay. So what happens in this case is that there are certain proteins. Let me show you first. As you can see here, As you can see here, for example, that this is the transferrin receptor mRNA, fine, and on this mRNA there are some iron response elements, right, in the 3 prime. So in the 3 prime we have some iron response elements, this folded area of the mRNA. So this area of the mRNA, if it is bound to the iron regulatory proteins, this green color, as you can see here, this green color iron regulatory protein, if it is bound, then the mRNA is stabilized, right? Means, again, let me explain, in the iron deficiency anemia, what happens is the iron is less. So if the iron is less, iron regulatory proteins which normally bounds the iron are free. So iron regulatory proteins, they will bind at the 3 prime of the this mRNA, which is called as iron response elements, IRE. And when there is a binding of IRE with the IRP, this mRNA is stabilized. And guys, when the mRNA is stabilized, this will be translated in the future. But guys, I don't want the ferritin mRNA. So during the low iron concentration, the IRP, okay, will bind the 5 prime region of ferritin, right? F for ferritin, F for 5, T for transferrin, T for 3. So 3 prime of the transferrin RNA and 5 prime of the ferritin mRNA. That is the site of binding or regulation. Okay, F and F, ferritin and 5. So IRE, the IRE will bind to the IRP, right, in the low iron concentration, and guys, this will cause a degradation of the ferritin mRNA. And similarly, in the high iron concentration, guys, the IRP will be bound with the iron and the ferritin mRNA will be active and will produce the ferritin protein, because once the iron is high, I need the storage protein. So hope you got it, right guys? So that is it. And this is in this side, again the alternative splicing has been shown, that from the 1, 2 and 3, 4 exons, I can just make a splicing, alternative splicing. I can use only 3 exons, 1, 2, 3 and 1, 3, 4, and that makes a different proteins. This is the isoform of the proteins I'll be getting. From this mRNA I'll be getting 1 protein, from this mRNA I'll be getting 1 protein, and from this complete mRNA I'll be getting 1 protein. So I can make multiple proteins from the same mRNA by the process known as alternative splicing, right? So that is again a important concept. Now guys, let me tell you one more point, that there are 2 terms that we are confused sometime, that what is the difference between microRNA and the short interfering RNA, right? That is siRNA and miRNA. So guys, the 2 or 3 important differences that we should know about them is that microRNA is most of the time, it is binding at the 3 prime UTR, as you can see here, and it is it is actually not binding completely. So there is no complementary base pairing, right? mRNA and microRNA are partially complementary, but siRNA is fully complementary, as you can see here, right? They are fully complementary. Second thing, microRNA binds at 3 prime UTR, but siRNA can bind anywhere. Okay. Third thing is that miRNA is most of the causing translational suppression, but siRNA, because it is fully complementary, it activates the agoproteins, which is called as a slicer, which degrades the mRNA. So these are the 3 differences that you should remember, the difference between miRNA and siRNA. And guys, siRNA is not most of the time derived from our own body. It is mostly from the exogenous sources, like for example the double-stranded RNAs of the viruses. Okay, so it is derived from the viruses. It is mostly exogenous, while the microRNA, our own body, our own genes will be producing. Okay, so these are the few differences that you should know, and that is why siRNA technology has been used nowadays for creation of the drugs, which are directly inhibiting the mRNA and doesn't allow the protein synthesis. Okay guys, so let's move to the next point. As you guys, don't forget these names, okay, during the process of microRNA synthesis. So during the process of microRNA synthesis, there are some proteins known as DROSHA, DROSHA, I have written here again. DROSHA is the example of the microRNA processor MCQ, PYQ. What is DROSHA? MicroRNA processor. As you can see, this is a primary microRNA and this is the pre-microRNA. So DROSHA is helping in the making of primary to pre. Then guys, there is a protein known as dicer. Dicer is a protein or the endonuclease which breaks the microRNA into multiple parts. As you can see here, this is the microRNA and dicer. Dicer has broken this microRNA to the microRNA which is almost mature. Okay, so these are the terms that you should know that I have already specified here: DROSHA, dicer and RISC. I told you the components of RISC, MCQ. Function of DROSHA, MCQ. Dicer, I told you it is cleaving the microRNA. So during the microRNA synthesis, there can be MCQ, which of the following is required or which of the following is not required. So this can be the MCQ again. Clear guys? So that is what we have to remember. So let's move to the next. So guys, what are the other processes that I was discussing? So I discussed about mRNA stability, right? Then now let's talk about microRNA that I have discussed already. Then mRNA editing. So our topic is post-transcriptional control, mRNA editing. Guys, they have asked till now only the examples that how the ApoB48 is synthesized from the ApoB100 mRNA. This is the example. So during the chylomicron synthesis in our intestine, we use the process called as mRNA editing. So that is the example of mRNA editing that you have to remember. Clear? Okay. So these are the points that I have discussed. So again, alternative splicing, mRNA stability, I have discussed microRNA and mRNA editing. Fine. So guys, this is the same thing. mRNA editing is shown here. So what you should remember is that what is the editing enzyme's name? The name of the editing enzyme is cytosine deaminase, MCQ. Previous year question, though it was asked long back, right, 6-7 years back, but this was the question, what is the name of editing enzyme? So cytosine deaminase, what does it do? It converts the CAA, a codon, to the UAA, right, which makes the stop codon. How the editing is done? By the creation of stop codon in the messenger RNA. So the protein which is formed will be now shorter, right? So that is how we do the mRNA editing. Fine guys. So now moving to the next point, that is translational control. Translational control is till now not being asked as such directly in the exams, but guys, one thing that we should know is that any protein which is destined to be part of endoplasmic reticulum, it has some signal peptide, and that signal peptide is being identified by, right, that is identified by a complex that is called as signal recognition particle. Fine. So as you can see here, that this is the signal peptide red color, this is the signal recognition particle pink color. So when this binds the translational stop, in the cytoplasm, and then it will go into the endoplasmic reticulum and the translation will resume again. So the proteins which are destined to be part of endoplasmic reticulum, they can be controlled like this translational control. So control is like outside in cytoplasm, the translation will stop by the SRP and signal recognition particle and the signal peptide binding. Guys, post-translational modification will be based on the enzymes. So that may be the allostric regulation of the enzymes by binding of some activators and inhibitors. Okay, so competitive or allostric inhibitors which prevents the enzyme activity. And what we can do is we can also alter the life of the enzyme or the protein. How? By the process of ubiquitin proteasome system. So guys, this is again a PYQ, ubiquitin proteasome system. What is that? This is a machinery in our cytoplasm which mark a protein for degradation in proteasome. How? See, it is like this. Let's suppose this is the pink color is the protein, and on this pink protein we will be adding a ubiquitin protein, right, as you can see by the green color. So ubiquitin protein will be added, and then this protein which has ubiquitin will be degraded by 26S proteasome. So this much you should know, that to degrade a protein in the proteasome, what we should do, we just add the ubiquitin proteins to them. That is how we do it. Clear? Fine guys, enzymes, definitely we know what is the allosteric activation and inhibition. So let's move to our next topic, mitochondrial genome. See guys, mitochondrial genome is again one of the super important topic that has been repeated several times in exams. So क्या पता होना चाहिए हमें mitochondrial genome, all the points, not a single point to be missed. Fine. Like humans contains multiple mitochondrial DNA genotypes. Okay, so one mitochondria has multiple genotypes or different varieties or sequences, that is heteroplasmy. How much is the mitochondrial DNA? Hardly 1% of the total human DNA. There are 10 copies in each mitochondria, and that is called as polyploidy. Okay, there is a very high rate of mutation, multiple times it has been part of MCQ. So the points are which I have highlighted, they have been part of MCQs or they have been directly the MCQs. Clear? Double stranded but circular, 16,569 base pairs, 93% is coding, right? Or you know, some book says or almost 97% is coding, and guys it lacks the intron, right? So there are no introns, there are 37 genes, and out of 37 only 13 makes the proteins. What proteins they are making? They are making the complex of ETC, like complex number 1, 2 and 4. Right, 1, 3 and 4, but not the complex 2. Succinate dehydrogenase is not coded by mitochondrial genome, but it is imported once it is made by the nuclear genes. Okay, then guys remember, that's why this is the line, and this line is somehow the favorite of examiner, that not all the proteins in the mitochondria are coded by mitochondrial genes. This point has been asked several times. This is the answer actually most of the time, that not all the proteins in the mitochondria are encoded by mitochondrial genes. So remember this point, this can be very important again. And this line again, high rate of mutation, has been part of questions many times as the answer. Guys, there is only one polymerase, that is polymerase gamma, which has all the activities like proofreading and polymerase, and also for the base excision repair. It has maternal inheritance, as we all know, mother is a powerhouse of our body, right, which is giving us mitochondria. There are some changes in the sequences of the genetic codes, and that is like UGA is not the stop codon in mitochondria, it is tryptophan. And guys, AGA is normally arginine in the nucleus, but here it is the stop codon, and AUA is methionine here instead of isoleucine. Fine. What are the names of disorders of mitochondrial genes? Like these are MELAS, that is mitochondrial encephalopathy, lactic acidosis and stroke. Then the MERRF and the Leigh syndrome. So there are multiple names, but you should know some names which are mitochondrial disorders, like these three. Okay. And guys, this is a pedigree chart that you can easily understand that only the females are, in the case of mitochondrial disorders, right, the female is transferring to all the babies, right, that is mother is transferring the disease to all the babies. Okay, but if the father has the disease, it is not being transferred. Fine. So let's talk about the next point. Guys, translation. So during the translation there are a lot of points are there, but the examiner has not asked much on the translation topic. Okay, but again, few points that I want to tell you is that for the translation process we need three things: mRNA, tRNA and the ribosomes. Okay, now there are various steps: initiation, elongation, ribosome translocation and termination of the process. So during the initiation process, what we need is 43S pre-initiation complex. So in the previous exams, what they have asked is that what are the components of 43S initiation complex. So that you should know. So that is again guys, the initiation factor 2 bound to the GTP, methionine tRNA and the 40S ribosome, that is a smaller subunit of ribosome. Okay, so these are the components of 43. Then we have 48, which is having mRNA, right? So there was a question like which of the following is not a part of 43S. So answer was, let's say, mRNA. mRNA is a part of 48S complex, and also the 4E. I told you the function of 4E that identifies the mRNA cap, right? And then finally ATS, guys, will be taking again the 48, then some GTP hydrolysis will be done, right? And 60S is also added, the larger subunit is added. Previously we took only smaller subunit, now we have taken the larger subunit also. Okay, so guys, this EIF2 which was having GTP, it is also called as GTPase, right? That is called as GTPase. Fine, and it is not mentioned here, but it is EIF5. Okay, guys, EIF5 is something that is known as GTPase activating protein here. Fine, so elongation and ribosomal translocation, we will see that. So finally this complex looks like this. This was MCQ, that where does the initiator tRNA with the methionine occupies the site. So guys, this occupies, because the first codon is always methionine, so it will occupy the P site. Okay, this is A site, P site and E site. So P site is being occupied, as you can see here, by the initiator tRNA. Fine. Now guys, in the elongation step, what we have to do is, we have to add the next we have to take the next codon, and the next amino acid carrying tRNA will bind here, like previously methionine, now phenylalanine is binding here. This phenylalanine and methionine, they will be added together with the help of the enzyme by making a peptide bond. So what is the name of that enzyme? This is 28S ribosomal RNA, which has peptidyl transferase activity. Which activity? So guys, this name is also a ribozyme. Examples of ribozyme, this name we should know. Enzyme which has peptidyl transferase activity is a part of 60S ribosome. Please write it. This is a part of actually the larger subunit, that is 28S ribosomal RNA. Okay, so that has a peptidyl transferase activity. Fine guys. Okay, so that is fine. So this is how what is called as a peptide bond is being made. As you can see, methionine and phenylalanine, they are bound together. Now guys, this can be again a question that ricin, okay, ricin is a poison from the castor seeds, and this can inhibit what? This can inhibit the peptide bond formation. So ricin from the castor is very toxic, right? And it has been used by Alia Bhatt in her movie Razi. When she attacked with the umbrella, she injected this poison into that soldier from Pakistan, right, that in the movie, and she killed him. So because there is no antidote for this. So basically ricin is found in the castor seeds. Okay, so elongation is done. We have made the peptide bond, so we are elongating the peptide. Now guys, the next point is ribosomal translocation. So guys, translocation means we will take the ribosome to the next codon, and this needs elongation factor number 2, and guys, this elongation factor number 2 is inhibited by the diphtheria toxin. MCQ again, right, has been asked. Diphtheria toxin inhibits the translocation step by inhibiting or by binding to the elongation factor number 2, eukaryotic elongation factor number 2. So that's how it acts as a poison, right guys. Then in the translation termination guys, once the stop codon is there, right, and the ribosome, A site of the ribosome will see the stop codon, and the releasing factors will come into the action, and they will release the peptide from the tRNA. Okay, so suppose this is a tRNA, right, and this tRNA is carrying the proteins, that is a peptide which has been formed. So this tRNA will release this peptide by the action of releasing factor, okay, by the action of the releasing factor, which induces the peptidyl transferase activity. That is a breakdown of the peptide bond. Fine guys. Now this question has been asked twice or thrice in exams, that is what is the importance of the wobbles hypothesis? See guys, we all know that genetic codes are redundant. Okay, what is the meaning of genetic codes are redundant or degenerate? That means that for the 20 amino acids, I have 64 or 61 codons. So obviously for one amino acid, I have multiple codons. One amino acid, multiple codons. Okay, similarly, one tRNA, which is an anticodon, can read multiple genetic codes. Okay, right? One tRNA can read multiple genetic codons. So that is called as Wobbles phenomena. So Wobbles phenomena explains, this was MCQ, answer is codon degeneracy. Again listen to me. Wobbles phenomena explains which process? Answer is codon degeneracy. So that's the answer. Right guys, so that is all. As you can see here, where will you see the codon degeneracy? This is a tRNA, and guys, in the tRNA we have an anticodon loop, right? So this tRNA is 5 prime to 3 prime. So then this mRNA must be actually 3 to 5, right? So the codon degeneracy will be seen at, as you can see, though it is not shown here, but you know guys, the third codon of the mRNA, that is 3 prime. Third codon, yeah, the third letter in the codon, and the first letter of the anticodon, right, at 5 prime, they will have the wobble base pairing, right? Like inosine in the tRNA can bind with U, C and A, okay? Inosine can bind with U, C and A. Is it clear? So that is all about the Wobbles phenomena. So guys, this is what we have covered is the most important points on translation process. Fine. Now let's talk about the recombinant DNA. So guys, we have covered most of the topics, right? This is the last topic, recombinant DNA. And this is again very important for INI-CET exam. So guys, what is a recombinant DNA? The recombination of the two DNAs. Like if I take a DNA of the plasmid, that is a vector, and the human DNA. And if I fuse them with the DNA ligase, that is called as the recombinant DNA. Clear? So what is the purpose and how we make it? So guys, one of the greatest discovery of the recombinant DNA was restriction enzyme. Restriction enzymes are present in the bacteria as a defense mechanism against the phage, right, bacteriophage. And you know what they do is they cut the double-stranded DNA of the phage, okay, at a particular site, right, that is 4 to 6 base pair long, and that is a palindromic sequence, right, palindromic sequence and 4 to 6 base pair long and double-strand DNA. So restriction endonuclease cuts double-strand DNA, 4 to 6 base pair long is a site where it cuts, and it is palindromic sequence. But guys, the point is, everything is written here, but this point was again asked in the MCQ, that bacteria prevents its own cut by methylation of the restriction site, okay, methylation of the restriction site. So means bacteria will prevent its suicide by the process of methylation process. Clear guys? Okay. And they're named like this, EcoR1 or something like that, like derived from E. coli. Guys, you can see GAATTC, this is a palindromic sequence. Palindrome doesn't mean from the English, यहाँ पे English वाला palindrome नहीं है, like CIVIC, civic, okay, CIVIC, so like that. So it is GAATTC, so this will be from 5 prime to 3 prime, and this will be from 3 prime, again 5 prime to 3 prime, it will be GAATTC, okay. So upper is 5 to 3, lower is also 5 to 3, same sequence, if I am reading from 5 to 3. You got my point? Okay, so that's how it is cutting. Now guys, when it is cutting, it is leaving some unpaired bases which are called as the sticky bases. So these are the sticky bases, okay. Now these sticky bases, if we take from the two organisms, if I take the sticky bases from the two organisms like plasmid and human, so these sticky bases, because they are unpaired and they are complementary, unpaired, complementary, see again, okay. Let's see again, that is, you can see these are unpaired and also complementary, A bind with the T, A bind with the T, like that. So if I take such two sequences from the two organisms, then I can fuse them, and that is called as recombinant DNA, right, as you can see here, okay. This is the insert, this is the cloning vector. So suppose this is a plasmid DNA, this is human DNA, and I am just cutting with the same enzyme like EcoR1, and then I'll be creating some sticky ends, and these sticky ends will fuse with each other using the DNA ligase, which makes the phosphodiester bond. Clear? Cutting with the same enzyme. That's why, site will be same, na? If I take EcoR1 from E. coli, the GWTC of the human and GWTC of the plasmid will be cut at the same location, same position. So that is how we will be getting the recombinant DNA. Fine, recombinant DNA, this is how we get. Clear, guys? Now, guys, there is a process that is called as cloning. Cloning means if I create the multiple copies of the same DNA, that is called as cloning. So nowadays we have PCR for that, right? Polymerase chain reaction. But cloning can be used also for the same purpose, though it is laborious process and not used commonly nowadays. But we should know as per the exam purpose. So guys, what we did is plasmid and human DNA was, human gene was cloned into the, let's say, plasmid, okay, which is a vector here. So this is a host cell. So now host cell has the, gene which is required to be cloned. So I want to make copies of a X gene, for example. I want to make the copies of, let's say, insulin gene. So I'll do that. So I have just transferred this into the host cell, and now when the host cell is multiplying and also the, the plasmid is multiplying, basically I'll be getting multiple copies of the same gene. You getting a point? So that is how we get multiple copies of the one gene that we can later on remove, identify and store them to make something called as library or the genomic library. Let's see some more points. See guys, there is a term that is called as a vector. Just listen to me. Vector means something which is a DNA carrier. So I want a DNA carrier. Now vector should have some properties, right? The vector should have, how to select a vector, like for example, plasmid is the most common, MCQ, plasmid is the most common vector used in genetics, right? Why? Because first of all, there are some features to that. That is, first, it has an origin of replication, right? Then it is a selectable antibiotic resistance marker. I can identify that which E. coli cell has the desired vector or the plasmid, which contains the gene of interest, that is insulin gene we have transferred. Okay, so they have a selectable marker that is called as the, let's say, antibiotic resistance gene. Okay, so these are the points that should be there in the vector. Okay, so as you can see here, this is a cloning vector. Cloning vector means it has an origin of replication, then it has a multiple cloning site and selectable marker. Remember these 2 points, origin of replication and the let's say the antibiotic resistance gene. Okay, so why we need this antibiotic resistance gene? Because I can identify the bacteria which are carrying the gene that I want to make copies of. Okay, why? Because that will grow on a antibiotic containing medium. That is why. Fine. These are the names of vectors, and plasmid is able to carry only 10 kilobases of DNA. So we can actually clone the genes which are smaller in the size. Phage, cosmid, bacterial artificial chromosomes and the yeast artificial chromosomes. So guys, the size is increasing, and some points we should remember, at least like plasmid is able to carry 10. Old question that cosmid is able to carry around 50 kilobases and has been asked cosmid. Okay. Guys, methods to transfer. See again, come back to the point. I want to make copies of, let's say, insulin gene, example. Okay. So first of all, what I did is I took the plasmid, I took the human gene. They were cut by the, let's say, same, at the same restriction site. They were joined by DNA ligase. Then they were transferred to the E. coli or the host. Now, how they are transferred? That's the point. So methods to transfer was a MCQ in the previous exams. And these 3 are the methods, or 4 are the methods, like electroporation, microinjection, gene gun, and incubating the cells with the calcium, right, under the low temperature. So that is how we are able to inject the genes into the host cell. Now guys, how to screen whether the vector, that is this vector, right, this vector which has been made, Okay, the clone that we have made, how are we able to identify that, how is it being transferred? So we have a process that is called as a blue-white screening, right guys? Blue-white screening, just name that you should know, is that screening the host bacterial colonies with the recombinant plasmids, right? So when the bacterial colonies are white in color, that means the successfully the vector has been transferred to the host, okay, which was carrying the insulin gene, suppose example, okay, just for example. So guys, this has been again asked 2 to 3 times in exams, the blue-white screening. It should be in your notes, right? Blue-white screening should be in your notes. Now moving to the next point, guys, there is a term that is called as recombinant proteins. Let's say, for example, some proteins like human insulin was created, okay, humulin was created. So that was created using a expression vector, right? Expression vector. So express means it will be expressed inside the host. So how to make it express inside the host? Because previously what we did was unable to make any protein because it was just carrying the gene, nothing else. We can make the copies but not the expression vector. So I'll talk about this. Guys, so that is what I was talking about, application of the recombinant DNA. So to make a protein inside the host cell like human insulin, what we need to do extra is, guys, we have to add a promoter which is identified by the bacterial RNA polymerase. That means bacterial RNA polymerase will identify ki, okay, this is my gene. Okay, it has the promoter, let's say the Pribno box example. So that is the promoter which is bound by bacterial RNA polymerase. So example, so promoter should be there. There should be ribosomal binding site so that it will be there in the mRNA. Okay, and that is the clone gene, suppose insulin gene or cDNA of insulin, right? So that is selectable marker. So guys, this is called as expression vector. So what is the difference between a cloning vector and expression vector? This can be the question. The expression vector which will make the recombinant DNA protein will have promoters and ribosomal binding sites. 2 points, please remember. Promoters identified by the RNA polymerase of the bacteria and ribosomal binding sites so that the mRNA which is formed from the DNA is able to bind the ribosomes of the bacteria or the host. Okay, and that's how they can make the proteins which is required. Clear guys? So this is called as expression vector. What are the uses of this technology? Recombinant DNA technology. So guys, we are discussing this point only. For example, humulin, growth factor, growth hormones, factor 8, erythropoietin, etc. Fine. So we are able to make proteins using this technology, but not very successful till now. But yes, these are the few examples. Clear? Then the other point is why to use mRNA instead of genes. See guys, during the recombinant protein synthesis, we don't take the genes, we take mRNA. Why? Because mRNA contains only the coding sequences. mRNA means obviously the mRNA which is in the cytoplasm, that means introns are not there. That is cap, tail, everything will be there. So we need a mature mRNA. So that has only coding sequences. Okay. And why we are not using gene? One more reason is that bacteria lacks the splicing process. अगर मान लो हमने जीन ले भी ली और उससे mRNA बन भी गया बैक्टीरिया होस्ट में, तो उसके पास splicing process नहीं है. So it will not be able to remove the non-coding introns. That is why. Okay, so this is again a question, part of question. And guys, this has been asked in the NEET PG also. What is the most important step in making the recombinant DNA proteins in the host? So the answer is the addition of high yield promoters to the cDNA insert. Okay, high yield promoters we have to use. Means the promoters which are strong promoters, like the bacteria which are producing the enzymes beta galactosidase. So such genes. Okay, so promoters we have to use for such genes. You got my point? So addition of promoter is the most important step in making the desired amount of human insulin, for example. So guys, why I have written the cDNA? Because I am using mRNA and I will convert the mRNA to the complementary DNA using the reverse transcriptase enzyme. Fine guys, you got my point? Second is site-directed mutagenesis. So guys, we will change the sequence of the DNA, right, to change the position of amino acids. Like for example, if I say lisproinsulin, that is proline and lysine is the normal sequence, but if I reverse it, 28 and 29, that is lisproinsulin. Okay, so site-directed mutagenesis can be used to understand the mutations. Okay, then next is gene therapies. So we can also use this process for the purpose of gene therapies, right? Like for example, beta thalassemia gene therapy, right? So we are using adeno-associated viruses are the most commonly used vectors in the gene therapies, right? So we are using vectors actually. Then construction of DNA libraries. Guys, I told you how to construct a library. Library simple here. Library means make the copies and store them. So we have two types of DNA libraries. One is called as complementary or cDNA library and other is called as the genomic library, right? So what is the difference between them? Because genomic library, suppose if the organism has a very small quantity of DNA, so I can actually clone the whole genome, right? So entire genome is closed, including introns and promoters. We use this reverse restriction endonuclease and the DNA ligase. Okay, so for the smaller genomes I told you, this cannot be expressed, right, because it has non-coding regions also, so it cannot be expressed, and it needs vectors with the higher DNA carrying capacity like bacterial artificial chromosome or the yeast artificial chromosome. While the cDNA library, suppose the genome is very large like a human genome suppose, so only expressed regions will be there, okay, coding regions. So we will use the reverse transcriptase, I told you, mRNA to cDNA. For a very large genomes like us, can be expressed, okay, because it is only expressing areas or the exons. Need plasmid, phage and cosmid. So carrying capacity of the vector can be small, no problem, right? Okay. Moving to the next point, that is one-liners that you should know, direct MCQs, okay. Number one, quantification of DNA methylation. So guys, the gold standard for that is bisulfite sequencing. That these are the things that we have to just remember. We can't do the details of that, and that is beyond our, and that is out of syllabus. I mean to say, मैं बोल सकता हूँ ये, ठीक है. But कुछ points हैं जो हमें सिर्फ नाम पता होना चाहिए और basic points पता होना चाहिए. जैसे कि ये वाला point, ठीक है. Bisulfite sequencing is a technique that is required for quantification of DNA methylation, like genomic imprinting detection. Okay, detection of histone modification. So guys, we do the chromatin immunoprecipitation, that is written as CHIP, chromatin immunoprecipitation. We can also do the mass spectrometry analysis. Guys, mass spectrometry is a technique which is again very important. Question has been asked twice in the exams, and this mass spectrometry is used for the screening and diagnosis of all inborn errors of metabolism, right? So that point, that is again you should know. DNA footprinting, guys, this is the again recent one and is an old question, right? It is required for identifying the protein binding regions on a DNA molecule, okay? Protein binding regions on the DNA molecule. So where does the RNA polymerase binds to gene? Okay, so how to know that? DNA footprinting, okay? So this is important, has been asked. So these first two has not been direct question till now. This one which I have highlighted has been direct PYQ or PYT. DNA fingerprinting. Guys, fingerprinting, they asked that what is the basis of fingerprinting. So the basis of fingerprinting is variable number of tandem repeats or the short tandem repeats. Now both in the option mark short tandem repeats because they are shorter and easy to analyze, right? That is why we have to remember that. Okay. So then next is number 4th. This is Number 5 is pyrosequencing. Guys, this is a method of DNA sequencing. So the DNA sequencing methods which have been asked in exams is mostly Sanger sequencing. But what is pyrosequencing? That is based on the release of pyrophosphates. Okay, so a sequencing method which is based on the estimation of pyrophosphates that is released during the process. Okay. Then Sanger sequencing, guys, Sanger sequencing again has been asked multiple times in exam, very important PYQ, PYT. But most of the time they're asking only one thing, that what is the principle. So it is based on the use of dideoxynucleotides, which contains no 2' and 3' OH groups. So they will inhibit the process of the amplification of the DNA at a particular point where the dideoxynucleotide is added. So guys, this is called as a chain termination method. So still this is considered as the gold standard for sequencing, but in if the next generation sequencing is available, that will be definitely the better one, but that is not available, right? Because that is still a process which is very costly and available in very limited areas or limited use. Fine. So what is the in the Sanger sequencing also we are able to analyze only very small amount of DNA, like only 700 bases per cycle, right? Next generation sequencing is also called as massively parallel sequencing, right, or sequencing by synthesis. Fine. So what is massively parallel means? If I do Sanger sequencing 1000 times at a time, I'm doing it for 1000 DNA molecules at a time. So that is called as multiple Sangers in one go. So that is actually next generation sequencing. Fine. It is actually very fast and more accurate, precise than the Sanger sequencing, though it is not easily available. Right, nowadays we have availability, but it is not frequently used. Okay, but it can, the whole genome of your organism can be sequenced by this process in just 1 or 24 hours to 48 hours. Okay, which will take much more time by the Sanger sequencing. So guys, you should know the name next generation sequencing or the massively parallel sequencing. Guys, remember one more thing, that to identify the point mutation, the sequencing is the best method. And if there is a question that which of the following is the best method to identify the point mutation, then mark the answer as the sequencing of the DNA. Okay. Microarray. Guys, microarray is also called as a DNA chip. And this DNA chip is very important question again. It has been asked that, it is based on the point that if I want to analyze, the genes from the two organisms or from the two part of our body, right? So what technique can be used? That is this process. So this is, I can use it primarily for the process purpose of comparative gene expression analysis. Remember this line, comparative gene expression analysis. I'll be using the point, that is this process that is called as a microarray analysis. Okay guys, fine. Fine, so let's move. So guys, this picture, especially in the right hand side, this side you can see here. So this is the picture, if you see, that is showing what? The DNA chip or the microarray analysis, where the green means the gene is more expressed in a normal cell, where the red means the gene is more expressed in a tumor cell or disease cell, and the yellow means the both the cells are expressing this in equal quantities. Fine. So again I am telling you, microarray is for comparative gene expression analysis, right guys? Okay, so PCR steps was asked in INI-CET. So that is first denaturation, that is separation of DNA, then annealing of the primers, and then the chain extension. So these are the 3 steps in the sequence, guys, don't forget them. Okay, guys, the Taq polymerase which is used in the process doesn't have the proofreading, but these are the 2 names, like from the Pyrococcus bacteria, PFU and PWO polymerase, that has proofreading function. Guys, recently there was a question that which of the following technique is best for identification of the mutations or detection of the low DNA concentration. So mutations which are present in the very less number of copies, or the very low concentration of DNA, if you want to analyze, right, which technique can be the better answer? Answer is digital droplet PCR. Okay, digital droplet PCR, right? So that we will convert the mixture or the of the PCR into the droplets. Each droplet will act as a separate, PCR, what I can say, PCR mixture. So I'll show you the picture also. So blue-white screening, guys, I told you. What is that? Blue-white screening. So then we have the pulse field gel electrophoresis. So this was once asked long back in some exam. Pulse field gel electrophoresis is required for the separation of large DNA molecules, like entire DNA, genomic DNA of a particular organism you can separate. So we will be using some pulse of the electric field here, which is keep on changing. So just remember the name, pulse field gel electrophoresis, right? I'm not talking of any point which is not part of the MCQ. Which is not previously being asked. So I am covering most of the points here. Almost 95% of the points that I am covering here are part of MCQs. Directly, indirectly, PYQ, PYT, whatever it is. Right? So I am not giving you unnecessary extra points in the last moment. Right? These are all the points. See, DNA footprinting was asked long back. It was again recently asked in just last 1 or 2 years. So there's nothing like, they will not ask it again. So that is why I am trying to give you every point which has been asked in the previous exams. Okay, so guys, this is pulse field. Page, polyacrylamide gel electrophoresis. Guys, this was again a question in some Central Institute exam. And what is the importance of that? That it has a very good resolving power. That it can separate the DNA molecules which are differing in just one base. Separate the DNA molecules which are differing in just one base per 1000 bases. Okay, fine. So that is one of the important point about the page. Fine. So they can accommodate also the larger quantities of DNA than agarose gel. So that you can ignore. Fine. Then guys, southern blot. To identify the DNA, what we have to do is we have to digest, that is restriction endonuclease digestion, break the DNA into pieces. Then separate the DNA using gel electrophoresis, then blot on the nitrocellulose membrane. We'll transfer the gel of the electrophoresis on a nitrocellulose membrane, so DNA will be there. Then we will just add the probe, which is complementary to your target gene. Suppose you want to identify the insulin gene from a mixture of genes. So you will make a probe which is complementary to the insulin gene. And step 5, hybridization, that is automatically happen, and washing, and then detection. Because the probe contains some dye, this will, glow, right? Western blot is for proteins, northern is for RNA, and eastern is for post-translational modification. The mnemonic, that is pizza and everything, like, roti, north mein roti, west mein pizza, P for protein, right? South mein dosa, D for DNA. Is it clear, guys? Fine. So next is fluorescence resonance energy transfer. So guys, this is required for protein-protein interaction, to study the protein-protein interaction, that is FRET. Okay, this was the point which was asked in exams. Okay, or we can also study the protein folding. Then we have the next point, that is circular dichroism. Guys, this is used to study again the protein folding, especially the secondary structure. And to study the 3D structure of the protein, we have the NMR and X-ray crystallography. Okay, NMR, that is nuclear magnetic resonance, and the X-ray crystallography. Remember these two, 3D structure of the protein, how to identify. So these are the 2 points. Guys, then we have FRAP, again has been asked. FRAP is fluorescence recovery after photo bleaching. So it is required for the mobility of, study the mobility of the fluorescently labeled molecules. Mobility, okay, fine. So molecules in the living beings. Just remember like coffee and frappe. Frappe means coffee. Coffee gives us the excitement or the mobility. Okay, so that is what we are using it for. Fine. Then guys, fish. Okay, Fish, the picture has been also asked. The basic principle is, we are using for this is that, using the fluorescence in situ hybridization. Fine. So fluorescently labeled probes will be used. And in this case, we can identify microdeletion, translocation, aneuploidy, right, like trisomy of 21 as shown in the picture. Right, as you can see, the trisomy is the 21st chromosome, which is targeted with a specific dye, right, or a probe, and we found the 3 copies of that, right. So trisomy of 21. Clear, guys? Then, guys, chip on the chip, that is chromatin immunoprecipitation plus microarray together, right. So what we are using it for? We are using for in vivo DNA binding sites for a given protein. So I told you that DNA binding sites, right, which are bound by a protein, we use DNA footprinting, but that was in vitro process. So this is in vivo, we can use it for this purpose, right? Okay, on a tissue sample directly we can use it. So that is chip on the chip process. Guys, RFLP is restriction fragment length polymorphism. So when you cut a DNA sample with the restriction endonuclease, what happens? We generate the different lengths of the DNA in different individuals like you and me. So why the different lengths are generated? They are generated because, the presence of variable number of tandem repeats between the restriction sites between me and you. So we have multiple variable number of tandem repeats at a particular site. Like if your DNA will be cut like this, and my DNA will be cut like this. So restriction fragment and the length is different between you and me. Why? Because the intervening DNA, which is variable number of tandem repeats, is having different length. Or it may arise also because of single nucleotide change. Because of the change in a single nucleotide, there will be loss or gain of restriction site. The cutting site will be lost or gained. Okay. Like for example, you can see here that this is a chromosome A and this is a chromosome B. Fine. So the cutting of the chromosome A is such a way that we'll be getting 2 fragments of 3 and 7 kilobases. Chromosome B from the person B will be cut in such a way that we'll be getting only 10 kilobase of the fragment. Right, guys? Okay. So that is like, the change in the length of the DNA because of the change in the site, restriction site. Okay, because of single nucleotide polymorphism it may be. Right, guys, you got my point? So that is how we'll be getting, different genotypes. Let's ignore that. You understood the process of RFLP and what is the basis of RFLP that you should remember, right? Guys, CRISPR-Cas9, was a recent Nobel Prize also in 2021, right? So CRISPR-Cas9 is a process, or the tool of genome editing, right? Genome editing means like deletion, insertion, etc. So that is CRISPR-Cas9 is a genome editing tool, MCQ. Then double strand breaks of the Cas9 are repaired by, I told you already, any type of double strand break. If they are talking, suppose they give the question that by using this drug in a cancer patient, there appears a double strand break, which will be repaired by which of the following process? So guys, your answer will be non-homologous end joining. So whatever is causing double strand break, the best answer will be non-homologous, commonly done in our body. I told you the reason. But if it is not given, and if they ask you mutation free or error free process, what our body should use, then it should be homologous repair mechanism, because that will be, replacing whatever is lost or whatever is incorrect. So homologous is best, but most common is non-homologous. So what our body uses? Non-homologous and joining. Is it clear, guys? Then this again was the question previous. So guys, these questions, I'm not just telling you that they may be only from AIIMS, that they may be from the previous areas like NEET PG also, DNB also, then from the JIPMER definitely, PGI definitely, AIIMS for sure, right? So techniques to identify the gene expression analysis. Gene expression means gene makes mRNA. So I should be knowing processes which can identify the mRNA, and the most important of them is northern blot, right? So northern blot, then I can use reverse transcriptase PCR. Okay, so that I can do that. Then microarray and RNase protection assay. So these are the 4. I'm just telling you 4. The list is long. But remember, at least northern blot, simple, because gene expression means gene makes RNA, and if RNA is made, obviously I can identify this using northern blot, right? So that's the process, guys. Okay. So fine, guys. So guys, in the end, I just want to tell you one thing only, that you're already in the process, right, of making your dreams true. So don't leave, or don't stay back, right? Step back, I mean to say, fine, because what you have already achieved, right, you should be proud of that, right? You are here, exam is almost reaching, right? And now what you need is revision, revision, and revision. Okay, so revise everything that is in your notes, and from the, for example, this revision session that you are studying in the LN-Next, or from any source, if you find some extra point which are PYQs or PYTs which are not part of your notes, add them, right? Don't leave them. Okay, so I hope this session was very high yield for you. So wish you all the best, guys. Okay, so I'm stopping this session now. All the best, do well, and let me know if you have any query. Fine, thank you so much, guys. Bye bye, all the best. बंद करो यार. हाँ, कितनी देर को हुआ यार? 2 घंटे का. 2 घंटे का.